Nursing 2533, Process of Human Diseases 1. This is Unit 2, the cell. So this is covered in chapters 4 and 5 of your book. A little bit of it is talked about in chapter 1. And what we're going to talk about today um, is the cell and cellular adaptation. So good idea to pre-read chapters 4 and 5 and then watch the rest of this video. So it really comes down to cellular adaptation. Almost all pathology is related to cells, and because nearly all diseases are the result of insult to cellular components. Insult to our stresses on a cell manifest into tissue, and then the tissue manifests into organs, which then organ systems, and then the organism. So it all starts at the cellular level. And when we talk about prevention, we have to prevent disease starting at this point. We have to either make the cells more resilient to the stresses placed upon them, or reduce the stresses placed upon those cells by whatever means that particular disease has. So it's also important to, for early detection of disease, to find the abnormalities when they're at the cellular level, because it's not widespread. It's not manifested to the entire organism at that point. So there's going to be a lot of definitions. In order to understand this, you have to know what a bunch of the words mean. So we're going to start out with atrophy. Now, atrophy is the decrease in the size or function of a tissue or organ. It really is about getting smaller. It's when the things atrophy, you don't get to use them as much because they're smaller or they're not functioning as well. Now, what causes atrophy? The most common cause is disuse. Now, before we get into that, I want to talk about different types of atrophy. There's basically two types. There's physiological atrophy, and there's pathological atrophy. The physiological atrophy is from a normal stressor, and oftentimes it's a normal course of physiology. For instance, post-pregnancy, the uterus will atrophy and get smaller again. It was enlarged greatly during the pregnancy, and it gets smaller postnatally because it doesn't need to be that big anymore. And a lot of what causes the growth is hormones, and so the removal of those hormones is what causes the atrophy. And this is normal and expected and wanted, really. Abnormal atrophy is when there's an abnormal stressor. And this all comes down to some sort of a loss of stimuli to the tissue that's involved. So this could be disuse because you're not stimulating, because you're not using it, and that's the most common thing. But it could also be blood supply. So ischemia is a huge cause of atrophy. When there's ischemia, there's not enough oxygen goes, not enough nutrients, so the growth not only stops, but it probably reverses, and you end up with some atrophy. We can have atrophy due to inappropriate endocrine stimuli, not enough endocrine stimuli. Most tissues need to have hormones to maintain, and when those go away, atrophy results. A loss of innervation is huge, especially motor neurons to skeletal muscle. When somebody has a spinal cord injury and is paraplegic, they get atrophy of the lower limbs because the motor neurons aren't stimulating the skeletal muscles, and when the skeletal muscles aren't stimulated, they shrink, they go away to a large extent. Normal functioning of motor neurons, the periodic firing of those neurons, as we learned in A&P, are important for muscle tone, and muscle tone is what prevents atrophy. So that's loss of motor neurons. So it's problems with innervation. Mechanical compression can lead to this, and decreased workload can lead to it. Decreased nutrition can do it. Like you've seen on the Save the Children commercials, those kids really have a lot of atrophy, especially of their limbs. People that are malnourished. If you see pictures of the concentration camp survivors and Auschwitz survivors and that, they are exhibiting lots of atrophy because of a general lack of nutrition. Aging can cause atrophy. Then it often does. Any sort of persistent cell injury can do it as well. So just using over and over again, like abusing a cell over and over again, will do it. So lots of causes for atrophy. Most common one being disuse. Hypertrophy is the opposite. It's the increase in the size of cells and their functional capacity. So hypertrophy also has a physiological and a pathological category. Physiological hypertrophy is normal stressors. So this would be things like exercise, building bulky and skeletal muscle. It's a normal course of physiological adaptation to use. The muscle cells get bigger. You don't get more muscle cells. The cells that you have grow more myofibrils and become more functional, more contractile, and they get bigger. So that's a normal type of physiological hypertrophy. There's also pathological hypertrophy. And pathological hypertrophy often comes from overuse, overwork. In this photograph here, it's a cross section of a human heart, and you will notice that the right ventricle looks normal, but the left ventricle, look at the myocardium. It's way too thick, and the lumen is way too small because of the hypertrophy of the cardiac muscle in the walls of the left ventricle. So that left ventricle is working to overcome the afterload. So this might be somebody with hypertension or like chronic hypertension or an aortic stenosis, for instance. So either one of those things will create too much afterload. It'll be too hard to push the blood in to the blood vessel. So Uh, so we have to increase contractility to make up for it. As a matter of fact, as this hypertrophy happens, there's less lumen to fill, less blood to actually move. So that actually decreases preload as well, which will decrease cardiac output. So to maintain the cardiac output at anything remotely feasible will take a lot more contractility, and cardiac muscle is muscle, so it hypertrophies due to this extra work. It's definitely pathological. As you probably noticed, this person is dead, and to dissect a heart like this, you don't do that out of a living person. So that's hypertrophy, the increase in the size of individual cells. Now, a similar thing is hyperplasia. Now, hyperplasia is an increase in the number of cells. So cell growth happens, like there's more mitosis. There is more cells in the organ or the tissue that makes up the organ. Now, hyperplasia and hypertrophy both result in an increase in size. The way you tell them apart, though, is microscopically. A pathologist will look at a sample of the tissue and decide that there are too many cells, and therefore it is hyperplasia, as opposed to the cells are abnormally large, which would be hypertrophy. And it's probably going to come as no surprise to you that there is a physiological hyperplasia and there's a pathological hyperplasia. So physiological hyperplasia is normal. It's normal stressors. It happens all the time. Cells grow in response. They grow in number in response to certain pressures. For instance, a physiological one would be breasts in pregnant women. They undergo hyperplasia. There are more cells, the breast enlarge as a normal course of pregnancy. Another hyperplasia thing would be if you move to high altitude, the increased erythropoiesis that would be caused by the lower partial pressure of oxygen is a hyperplasia, an increase in the number of red blood cells. Normal, acceptable, as a matter of fact, it's necessary. Now, an example of pathological hyperplasia would be increased adrenal gland size due to increased ACTH, adrenocorticotropic hormone. This could be caused by a paraneoplastic syndrome. Sometimes, like a lung cancer, will start to release hormones, and a big one that they release is ACTH. The tumor itself starts to act like the pituitary gland, releases ACTH, and the adrenal cortex responds to this stimulus and ends up enlarging, it's hyperplasia. This is not a good thing. So hormonal stimulation can cause it. Increased physiological demand can cause it. Chronic injury can cause it. An example of a chronic injury that causes hyperplasia is a callus. You are rubbing your skin in the same spot over and over again, and you end up with a hyperplasia of the epidermal cells in that spot. And that's what a callus is. Now, oftentimes, because the things that cause hypertrophy overlap significantly with the things that cause hyperplasia, oftentimes tissues react with both, that you'll increase the number of cells and the cells that you have will increase in size. And that's, uh, it blurs the line a little bit between hyperplasia and hypertrophy. This is a pathological hyperplasia here. You'll note that the photograph on the left is a normal skin. There's the dermis, there's the epidermis, the stratum basale is around here, the stratum corneum's here. It's a stratified squamous epithelium. This is a photomicrograph of the same magnification of a pathology of the skin. And look at the thickness of this epidermis here. And this incredible amount of hyperplasia. If you count the number of cells that are in this area versus the number of cells that are in this area, there's a lot of hyperplasia. And what this is psoriasis. This is plaque psoriasis, which is a pathological hyperplasia. Okay, we're going to shift gears a little bit right now. We're going to look at differentiated tissues, and we're going to define differentiated tissues. Now, we talked about this in AMP1, so it should be just a quick review here. But there are 4 types of tissues, but all those tissues come from the same original cell. At one point, you were just one cell, and then 2 and 4 and 8 and 16 and 32, etc., etc., etc. And those cells in that early stage, in that it's not even an embryonic stage yet, it's the blastosphere stage, those cells are undifferentiated. They're all about the same. They have the same genetic makeup, and all they're really doing is reproducing. And then they start to differentiate. They become different things. At first, it's endoderm, ectoderm, and mesoderm, and then that develops, and we end up with 4 primary tissue types, and that are completely different than one another. So the first type is epithelia. We've talked about this a lot in AMP. Epithelia is the covering and lining tissue, and it's glands. So it's skin, mucosa, endothelium of the blood vessels, that kind of thing. It's all that covering and lining or glandular tissue. It is highly cellular. There's not very much extracellular matrix, and it forms the barrier between 2 different environments, and therefore it takes a beating. So because it takes a beating, it gets replaced a lot, and so it's very highly mitotic and has a high metabolic function. Another type of tissue is connective tissue. This is the most common tissue type. It's loose and dense connective tissue, bone, cartilage, adipose, blood. So there's dense regular, dense irregular. All of that stuff is all connective tissue. And what it has in common is that the vast majority of the bulk of it is extracellular matrix. There are relatively few cells. And the cells that are there are very different than, for instance, the epithelial cells. Muscle cells are contractile, excitable, elastic, and extensible. Nervous tissue includes all the excitable neurons plus the glial cells found in the central nervous system, the peripheral nervous system, the autonomic nervous system. Uh, all of that is nervous tissue, and those cells are different than the other tissues, than the ones found in the other tissues. Now we need to uh, just have taken this review because the next few definitions are going to depend on understanding that. Now, within each one of these primary tissue types, there are the subtypes. So, for instance, in epithelium, there is uh, a stratified squamous and simple columnar and all of those things that we talked about in AMP. And those cells are differentiated from each other because they look different and they behave different and they're there for different reasons. And function and form, as we know from the principle of complementarity, follow each other. So what happens if the function changes? We get something called metaplasia. Now, metaplasia is the conversion of one differentiated cell type into another. Now, it's not the individual cells that change, but their progeny change. And over time, this metaplasia happens. Now, metaplasia, by definition, is reversible change. So this is an adaptive response that cells have to chronic persistent injuries. So what happens is the cells become the type of cell that gives the best protection against that injury, that insult. So there's all kinds of examples of this. In smokers, glandular epithelium of the pneumocytes, of the cells of the respiratory system, get replaced by a squamous epithelium. So the glandular epithelium gets replaced by squamous epithelium, which is got an entirely different physical characteristic, but it is more resistant to the persistent insult of the tobacco smoke. And so we say that tobacco smoke leads to a squamous metaplasia. We get this same sort of thing happens at the cervix, at the endocervix, at the uterus. The mucosa of the vagina is a stratified squamous epithelium because it has to resist friction. But the cervix needs to secrete mucus, so it needs to be a columnar epithelium, or pseudostratified at least, because it needs to have goblet cells. Now, if the cervix itself starts getting a persistent injury, say from an infection, like genital warts, the HPV virus, then what happens is the cells start to become squamous, and you lose the ability to make the mucus that is necessary, but we gain the ability to protect against that persistent injury. So it's a trade-off. And this is what they're looking for in the pap smear. When you get a pap smear done and the results come back and they say, oh, there are some abnormal cells, what they're saying is that metaplasia is taking place. So metaplasia, although it's reversible by taking away the persistent injury, is the first step towards irreversible change. So here is some epithelium, and you'll see that right along here, this is the good epithelium. This, see, it's a columnar epithelium along here, and then it's turned into this locus of stratified squamous is what it's trying to be, and I mean that's quite a large collection of metaplasia. Now, if the injury goes away, if the persistent insult goes away, then eventually these cells just die and are replaced with these cells. And this will be reversible and it will go away. However, if the persistent injury increases or stays persistent, then maybe these cells will, when they get replaced, will get replaced with cells that look more like this, and this will grow. So this is really the cells responding to changes in the environment and adapting to the new stressors. Now, dysplasia is the next step in this, and this is disordered growth. So it loses its organization, and you get cells that will vary in size and shape, and the nuclei tend to get enlarged because the cells don't know what to do, so they try and do everything. We get irregularities. They stop having proper arrangement because they're trying to change as an adaptation to a persistent insult. So here's a photomicrograph of some dysplasia. You'll note that over here on the left-hand side, this is a normal stratified squamous epithelium. But when we get in here, to the right, this is dysplastic. This is dysplasia. Look at the size of the nucleus here and here, and contrast that to the size of the nucleus all through this tissue. This is actually quite extensive dysplasia. It's lost its organization. See how there's almost layers here, and this is just a hodgepodge. So this is dysplasia. So this brings us to the idea of carcinogenesis. Carcinogenesis is really the process that cancer cells are formed, and they form from normal cells. There's really good theory, and most cancer research now is being targeted at the idea that metaplasia and dysplasia are the first steps on the path towards carcinogenesis, that somehow the carcinogens are causing cellular damage. The cells are reacting first metaplastically and dysplastically, and then they go further, and so these normal cells become cancer cells. And really what they're trying to do is they're trying to become stem cells to replace tissue with tissues that are better at protecting. And so it backfires. It misfires. The dysplasia gets carried away. The cells lose any semblance of their differentiation. So we call this loss of differentiation of the cells anaplasia. Ana is apart, so a growth apart. Anaplasia. So it's the loss of differentiation of the cells, their orientation to each other, and when we start to see these anaplastic cells as opposed to metaplastic cells, now we've crossed the line and we're into tumor type tissue. Now in here This is an anaplastic cell. You'll notice that the nuclei is gotten huge and that it's lobed. It's almost like there's multiple nuclei in here all overlapping each other. Um, it's undergoing mitosis so quick that there's not enough cytoplasm to divide. It's starting the next mitosis even before the this cytokinesis can happen. Um, so that cell there is anaplastic, and it's well on its way to becoming a tumor. Now, hopefully at this point, the natural killer cells or the cytotoxic T lymphocytes will remove that cell, and that will be the end of it. But if that cell is reproducing and it's sending signals to the surrounding cells, they may uh, follow along. There's probably some metaplasia happening elsewhere. It looks like maybe right up in here, there's a few things going on as well. So this is kind of the first, like this is where it starts to cross the line, this loss of differentiation. Now, when the anaplastic cells start to form a tissue, we call this a neoplasm. This is an autonomous growth of tissue. And there's no normal restraints on the cell proliferation. There's all kinds of anaplasia within the cells. There's dysplasia going on. And the cells kind of lose their cell life cycle. They don't have their programmed death. They undergo mitosis very, very rapidly. So neoplasms are very metabolically active and they're undergoing mitosis a lot. So they almost go right from telophase into the next prophase very quickly. And they kind of skip G1, S, and G2, or they abruptly shorten them at least. So another definition, neoplasm. So an autonomous growth of tissue without normal restraints on cellular proliferation. Neoplasm, new growth. Okay, so we've talked a number of times leading to metaplasia, dysplasia, anaplasia about injuries and insults to cells. So what are some of those insults? Probably the most important one, and the one that is the most common, is ischemia, hypoxia. Ischemia is really a loss of blood supply. Hypoxia is a loss of oxygen to the tissue. Usually, ischemia causes hypoxia. There are some other causes of hypoxia, but we won't go there. Things like carbon monoxide poisoning and that kind of thing. But usually, it's got to do with ischemia, with a lack of blood going to the area for blood clots or whatever. Could be loss of blood pressure, shock, that kind of thing causes ischemia. Another injury would be a nutritional injury where you're not delivering the nutrients that the cell needs in order to do. So like a hypoglycemia would do it, or not enough vitamin C, so that will result in scurvy, and that's a nutritional injury to the cells, and then the cells try and react to it. They try to adapt to that. There's a lot of infectious injury to cells, especially viral. Viruses enter cells and they hijack the translation transcription protein synthesis mechanisms, and that injures the cells. When we talked the immune system in AMP, we really looked at it fairly closely, and we talked about NK cells and cytotoxic T lymphocytes injuring cells. They get rid of cells. Sometimes complement. Inflammation will cause cellular injury. Um, there can be chemical injuries. Chemical injuries can be from ingested substances, splashed on tissues, anything like that, and changing the pH, introducing poisons, that kind of thing. Chemical injuries, and not the least of are mechanical injuries, physical injuries. Um, Remember, like you fall out of a tree and get a contusion and a bruise and break a bone, those are going to injure all the cells involved. Uh, so we're not going to talk a lot about the nutritional injury, and we're going to talk about the immunological things in Unit 3. But today we're going to talk about ischemic cell injury, because this is important, and this is really important to understanding some of the pathologies that are going to follow in this course and in the next course. So like when we start talking about myocardial infarctions, it's an ischemic injury to the myocardium. So what happens? Well, if we have ischemia, then the cell will not get enough oxygen. When the cell doesn't get enough oxygen, it can't make ATP. And up to 70% of the ATP that a cell makes is used in the sodium-potassium pump. And the sodium-potassium pump is used to maintain electrochemical gradients. It's pumping sodium out of the cell, potassium into the cell, and setting up osmotic gradients. And so the whole chapter that we did in A&P on fluid and electrolyte balance, pH balance, really is lost here. We talked when we talked about muscles, and then all in AMP1 when we did the nervous system, we talked about resting membrane potentials and excitability of cells and how it depends on sodium potassium pump. That is what we're talking about. No ATP, no, it's a sodium potassium pump. There's too much sodium in the cells. But there can also be too much calcium in the cells, because what happens is when the electrochemical gradients break down, then the things that keep calcium out kind of break down as well, and calciums move in. Calcium ions move in. When calcium ions move into the cell, and generally the cell has very low intracellular concentrations of calcium, when it comes in, it interferes with the mitochondria. And the mitochondria are the main source of ATP. And then that exacerbates the problem because the mitochondria aren't making ATP, and the electrochemical gradients get even worse. There's no more energy to maintain cellular structures. And the plasma membrane and the nucleus and a lot of the structures start to degrade. And this can lead to cellular death. This is a very common cause of cell death. So, kind of to recap, cell injury is often caused by ischemia, and that's the interruption of blood flow to an area. And that results in hypoxia. That's probably the most important thing. But it also results in an accumulation of metabolic waste, because the blood not only brings things to the cells, it takes things away. So the metabolic waste isn't taken away, and the cells start living in a soup of their own metabolic waste, and that can lead to injury and maybe cell death as well. And then not the least of the interruptions is to nutrients. The cells don't get delivered the nutrients that they need, even in glucose, for instance. So Decreased oxygen means decreased ATP, which means less sodium potassium pump, right? And not only does that mean difference in electrochemical gradients, but it also means a difference in osmotic gradients. Back in the lab in AMP1, we did a demonstration where you put a, a tube of dialysis with uh, with a concentrated solution in the tube, and you put that in reverse osmosis water, and the tube swole up because the osmosis drew water into the little sausage thing that you had, and it swole up. Well, if the sodium that's normally pumped out ends up staying in the cells, that causes an osmosis. The inside of the cell starts to become hypertonic compared to the interstitial fluid. And normally it would be the other way around, or it would be isotonic at least. This, this kind of hypotonic extracellular versus intracellular means that water will move into the cells by osmosis and the cells will swell. And we call this hydropic swelling. So this hydropic swelling then really disrupts a lot of what's going on in the cell because the cell is being stressed and swollen. It stresses the plasma membrane and things. Now, at the same time, intracellular calcium levels are going up, and this disrupts the mitochondria, as I said before, which then results in even less ATP production. So how does the cell get any ATP? It does it anaerobically. But when you do an anaerobic respiration, you only get 2 ATPs, and you end up with lactic acid. And so the lactic acid builds up intracellularly, and that lowering of the pH affects the tertiary structure of the proteins that are in the cells and makes the proteins non-functional. It damages the plasma membrane. It damages the mitochondria. It can lead to cellular death from there. Now, you would think, okay, this all sounds pretty bad. But there's a paradox here, and it's something as a practitioner you're going to need to be aware of. You would think that restoring the blood flow would be a very good thing. But there's a couple of problems with that. When you restore blood flow to an area, that blood brings even more calcium to the area because blood has fairly high calcium levels. And oftentimes that extra calcium is enough to trigger something called apoptosis, which is cellular death. The extra calcium will increase intracalcium levels even further and trigger this. Now, the other thing that happens is if there's mitochondrial dysfunction, reintroducing oxygen doesn't mean that all of a sudden those mitochondria will start functioning because they're dysfunctional because of the calcium. This extra oxygen doesn't get used in Krebs cycle and an oxidative phosphorylation. So this extra oxygen ends up becoming free radicals. And free radicals cause inflammation. And inflammation causes cellular damage, and the cellular damage causes tissue damage and organ damage. So we can end up with a severe inflammation because of this. So it's not a simple matter of just restoring blood flow. Sometimes restoring blood flow can be bad. So here's a schematic diagram just to sum this up. So if there's something blocking an artery, then you end up with ischemia. Because of the ischemia, you get decreased ATP, Krebs cycle, etc., decreased aerobic glycolysis. Because of the lack of ATP, the sodium potassium pump turns off. There's more sodium in the cell. We have an increase in anaerobic glycolysis, which leads to lactic acid, which then screws up the hydrogen ion, so it becomes more acidic. The pH gets lowered, which then brings more sodium in. Uh, the sodium and calcium get exchanged because the sodium comes in, calcium drives in to get away, and blah, blah, blah. That calcium screws up the cell membrane because it changes the enzymes that make phospholipids, as does the hydrogen from the lactic acid. So the cell membrane gets damaged, and this stuff goes back more and more until the cell just dies, and that's called necrosis. Now, it doesn't necessarily go that far. There's a tipping point. There's a point where if the insult stops, the cell can recover. So we call this damage reversible cell injury. It's acute, right? And it can change given changes in circumstances. Now, one of the most important reversible cell injury things is this hydropic swelling. So it's an increase in volume. It's caused by different toxins, by viral infections, by ischemia, heat or cold, anything like that causes this loss of the sodium potassium pump can do it. Um, it it involves the control of sodium. So that's keeping sodium out by the plasma membrane and the sodium channels, the sodium potassium pump, and the fuel to run the sodium potassium pump, the ATP. All of those things have to be intact to prevent hydropic swelling. Now, if the cell does get hydropic swelling and a supply of ATP goes back up, the sodium potassium pump can correct that, the plasma membrane can repair, and the cell injury has gone away. Here's a photomicrograph of hydropic swelling. You can see that these cells in here, and this would be an area of ischemia, are all swollen. The cytoplasm is huge compared to the neighbors out here. Now, another thing that can happen in cells is accumulations of stuff in the cells, of intracellular materials, and that will impair the cell functioning for as long as that's there. You get rid of the stuff and the cell can come back to normal. So there's all kinds of substances that can accumulate in cells. Some of them are just normal components of the cells. Some of them are abnormal. Some of them come from within inside the cells. Some of them come from outside the cells. Some of these accumulations can be quite harmful. Some of them can be innocuous, and it kind of varies. It usually has to do with some sort of faulty metabolism of the cell. The cell isn't processing things correctly, or there's some biochemical processes that are happening that are related to the pathology, like various toxins can cause this accumulation. So there's a couple of different types of things that accumulate. One is nutrients. The two big ones that matter are fat and glycogen. But you can get vitamins, minerals. All these things are normally stored. If you get excessive storing, then they accumulate. It's called abnormal accumulation. So an example of this would be in chronic alcoholism. The hepatocytes don't uh, don't metabolize lipids correctly. So the intrahepatic lipid metabolism is disturbed, and fat starts to accumulate in the liver cells, and we call that fatty liver. It's often a sign of alcoholism, but it can be a sign of liver damage from a number of other things. Sometimes um, hepatitis can cause this kind of thing, or a use of a chronic use of painkillers can cause it, or various other medications that people are on can lead to fatty liver problems. But this is an abnormal accumulation of fat in the hepatocytes. Another place that we see this a lot is the accumulation of cholesterol. Like a little bit of cholesterol is necessary, but too much cholesterol starts to accumulate in the endothelial cells of the cardiovascular system, and we call this atherosclerosis. The cholesterol actually accumulates within the cells. We can have accumulations of glycogen um, for various reasons, uh, because we're not breaking the glycogen down properly. The fat soluble vitamins you can overdose on them and they get stored. Things like vitamin A are particularly dangerous. Vitamin E. Various minerals can do that as well. So some of these things are problems in metabolism. So these metabolic issues, this an inability to metabolize, could be because the thing is just it just can't be metabolized. So these would be exogenous such things like so dust, carbon dust, or silica dust from you know will accumulate in the lungs. So silica dust uh, it happens in minors. Both my grandfathers died of silicosis because they both worked underground and they breathed a lot of dust. And there was an accumulation of silica, basically sand, quartz, in the cells of their lungs, and they died of lung disease because of it. Carbon from smoke inhalation will accumulate. So that could be cigarette smoke, but it could be from environment, that kind of thing. Sometimes there are missing enzymes. Uh, that means that endogenous things don't get complete metabolism. There are people that have storage diseases, and it's hereditary. It's got to do with a faulty gene that means that they don't make the enzyme that's necessary. There's a common one where well, relatively common one, where people can make glycogen from excess glucose, but they lack the enzyme that allows glycogenolysis, which is breaking the glycogen back down into glucose when it's necessary. So they just store glycogen and they can't take it out of storage, and their muscle cells and their liver cells and the cells that are storing glycogen end up with an abnormal accumulation of glycogen. These are called hereditary storage diseases, and can happen to a number of substrates as well. Now, a third thing that can happen is abnormal proteins, because the proteins in the cells have to have a specific shape and size for them to be functional. And if you're making the proteins and they're abnormal and they're non-functional, then the signal to keep on making more will continue because you're not satisfying the demand, because the proteins that you're making are incorrect. Now, a couple of diseases that are involved with this would be things that have to do with prions. Uh, so that would be like mad cow disease, uh, Jakob-Creutzfeldt's, Kuru. Like these diseases are neurodegenerative. The neurons seem to be susceptible to these abnormal proteins. There's another protein called the tau protein that is being identified in the cortex of the cortical neurons of the brains of people with Alzheimer's. Now we're going to talk about Alzheimer's in the next pathology course, not in this one, but there's this accumulation of these tau proteins, and they're called the neurofibrillary tangles, this faulty accumulation. So this sums it up. So we can have abnormal metabolism leading to accumulation of metabolites. We can have inappropriate proteins, so protein mutations, proteins not being made properly, so they're not folding properly, and so they accumulate because they're not doing the job that they want. We can have a lack of an enzyme, so the substrate builds up and instead of the product. So some of these things cross over. So, for instance, glycogen would fit into this and this, or it can be the ingestion, or like that includes breathing of indigestible materials, and they accumulate as well. This is a photograph of carbon accumulating in lungs. This is a photomicrograph of accumulation of cigarette smoke in lungs. So when we look at this abnormal accumulation and we look at this hydropic swelling, we're going to see structural features in the cell. And it's going to be noticeable when you look at the cells, or if you look at the cells. So we're going to see things like, because of the swelling, we're going to see an increased intracellular volume. So the cell membrane is going to bulge out in places. We call these blebs. The mitochondria will swell and they'll lose their shape, and a lot of times they lose their function. The endoplasmic reticulum is susceptible to this extra fluid, and it will start to break up and turn into vesicles. They'll dilate, and the membranes of them will lose their order. And as it does that, it releases the ribosomes that are on the rough ER, and the ribosomes become disassociated with the ER, so they're disaggregated. They're not attached, and they're floating around inappropriately in the cell. But this is, at this point, still reversible. At some point, it reaches a tipping point, and nobody really knows where that tipping point is. But when it reaches that, the cell will die. The cellular injury is too much to overcome, and the cell will die. And it dies in one of two ways: necrosis and apoptosis. Now, of the two, you want apoptosis. Necrosis is an unprogrammed death. It's when the cell dies inappropriately, unexpectedly. So what happens here is that the cell and the organelles within the cell will swell up due to the water coming in, the hydropic swelling. ATP will become depleted because the mitochondria aren't working properly. The plasma membrane will be stretched and become increased in permeability. The cell will die, and as the cell dies, it kind of bursts, and the contents of the cell end up in the interstitium, and this results in inflammation. And the problem with acute inflammation in the area is that this causes further cell injury to the neighboring cells. And oftentimes these cells may be in that, still in the reversible stage, and they'll get put over into the irreversible, and the necrosis will spread. The cells will spread, like cell death will spread through the tissue. So when the cells die, they die in 4 types of necrosis. One is called coagulative necrosis. Coagulative necrosis is usually what happens when we have ischemia, which is the most common cause of cellular death and necrosis. So therefore, coagulative necrosis is the most common type of necrosis. What happens is the nucleus breaks down and is degraded. Sometimes it's extruded, it's out. So the cell has no more nucleus. So for all intents and purposes, the cell is dead. The cell has got the swelling, the hydropic swelling, the disaggregated ribosomes, all of that stuff, the plasmabrebs are all present, but the cell doesn't burst right away. It happens later. The intra and extracellular lytic enzymes then cause it to disintegrate. But the cell is dead but still there in coagulative necrosis. Now, we'll contrast that to something called liquefactive necrosis. And liquefactive necrosis, the cells liquefy, they dissolve, they just are gone. And when they're gone, we get something called an abscess, a cavity formed in the tissue because of the amount of cellular death. And it causes really abrupt inflammation, which then puts even more cells into a necrotic state. And so this thing will grow, and it's a space filled with cellular debris and pus, really. This often occurs in the brain post-stroke. As a matter of fact, when I was a student in my anatomy course, I was assigned a cadaver that I dissected from Labor Day until the 24th of May. When we took out his brain, we counted the abscesses in his brain and figured out that he had had at least 15 strokes. His brain was a mess. It was like Swiss cheese. There were these big gaps in the neural tissue. And it was caused by liquefactive necrosis, post-ischemia. There's another type of necrosis called fat necrosis. This is when adipose tissue has a trauma. Pancreatitis causes this a lot. And there's these little white areas in normal adipose tissue. They're like little chalky, calcified little nodules. And that's dead tissue, so it's necrotic. The fourth type is something called caseous necrosis. And caseous necrosis, caseous means cheese-like. So it's a characteristic of tuberculosis. Now this happens when the tuberculous bacillus causes a necrosis surrounding it, and it kind of encases the infection. And the tissue is dead, but it's not coagulative necrosis, but it also doesn't disappear. So it's not liquefactive. It's something in between. And it ends up with like a cheese-like texture. And that's why it's called caseous necrosis. The big thing for that is tuberculosis. So when we look at this, there's some photographs of things. This is coagulative necrosis. See how the toes are black here? But they still look like toes. The shape, the outline of the tissue is intact, but the tissue is dead. This is likely a diabetes. At this point, it's actually gone to gangrene. We'll talk about that in a minute, but the point here is in coagulative necrosis, the tissue looks like it's intact because the cells are still taking up the same amount of space. This one is a liquefactive necrosis. This is the abscess in the brain post-stroke. You can see it's just an open cavity. And when I said Swiss cheese, I literally meant that. These little white specks in here, this is a pancreas. This is post-pancreatitic fat necrosis. This is caseous necrosis in lung tissue involved with tuberculosis. So if we get this necrosis and it spreads through large parts of the tissue, then this is called gangrene. And so it's cellular death in large amounts of tissue. And it's usually because of ischemia. That's the biggest cause. And there's a couple of different types of gangrene. There's dry gangrene. Dry gangrene is coagulative necrosis. It's this black and dry, wrinkly tissue, and it's sharply demarcated between the tissue that's still alive and the tissue that is dead. If we go back to this slide, you'll see that there's a line here where this is gangrenous and this isn't yet. This is gangrenous, this isn't yet. It may become, the gangrene can spread, but so this would be dry gangrene. Now, wet gangrene is when it's liquefactive necrosis, overtaking a whole organ. And so it normally happens in internal organs. It doesn't so much happen in the periphery, like dry gangrene does, because of this whole kind of softness of the organs. So sometimes, like the pancreas can undergo a liquefactive necrosis and just basically disappear. Now, gas gangrene happens when there's an infection by an anaerobic bacteria. The big thing for this one is Clostridium. You probably have heard of C. difficile, which is Clostridium difficile. And one of the things about Clostridium is that they like to eat this dead tissue. And so, and they don't mind the lack of oxygen because they're anaerobic. They make their ATP anaerobically. And so one of their byproducts is gas. And so basically this necrotic tissue gets filled with bubbles. And this is called gas gangrene. So the other type of cell death is called apoptosis. Now, apoptosis is normal. It's programmed death. If you think of apoptosis as cellular suicide, necrosis is cellular murder. So apoptosis is really the cells, when they outlive their usefulness, go out on an ice floe and stop being a burden on the group. They go away. Now, in a cell's life cycle, there's a constant balance between the the need to keep on functioning and the need to commit suicide, to go away. And there's this balance between the anti-apoptotic signals and the pro-apoptotic signals. And in normal, healthy, living young tissue, the anti-apoptotic signals predominate. As tissue is damaged or ages, or as the cells age, then they stop responding to the anti-apoptotic, and the pro-apoptotic signals start to predominate. And when that happens, the cell dies. And we need this. We need this to get rid of cells that are no longer any good, cells that are infected. Cells, so when we talk about killer T cells killing an infected body cell, what they're in fact doing is they're attaching to the infected cell and increasing the pro-apoptotic signals that that cell is getting, and then the cell dies. It commits suicide. Old cells that are obsolescent get removed. It's a way of preventing overpopulation. It's really no different than normal death by old age in humans. If everybody stayed alive too long, then we'd have huge overpopulation issues. So an organ needs its normal architecture, needs its normal size. We have to remove the older cells and replace them with new ones to maintain function and architecture. Mutant cells, cells that are not doing what they're supposed to do, usually undergo apoptosis. They're gotten rid of. Now, the huge difference with apoptosis is it's programmed, it's timed, and it's controlled, so therefore it doesn't cause any inflammation. And this is huge because it doesn't spread then. So what happens is these dead cells just get phagocytized, usually by macrophages, and they're removed without any inflammation. Now, one of the big triggers of apoptosis is intracellular calcium levels. And when intracellular calcium levels go up, then mitochondrial injury happens and all that stuff that we talked about. When it reaches a certain point and it's irreversible, then that signals apoptosis. There's a protein that's involved, and we'll talk about this more when we're talking about cancer, but there's a protein called P53. And what P53 does is when there's a problem in DNA and the DNA is damaged, it halts the cell life cycle for long enough that repair proteins can repair the DNA. So P53 kind of buys the time to repair the DNA damage. If the DNA is damaged beyond repair, then the P53 triggers apoptosis. And so it's basically almost like a decision maker. It's like, okay, we're going to see if we can fix this. Oh, no, we can't fix it. Away we go. It's time to leave. And P53 is involved with this. You can Google it. You can do a lot of things to read about it. But that's in essence, that's the Reader's Digest of what it does. Now, I've already said T lymphocytes can trigger apoptosis as well. really apoptosis is the default pathway. It's cells are going to undergo apoptosis, or else we would be just huge. Like the cells have to be removed. Um, the thing that prevents it is the anti-apoptotic signals. And in a big way, healthy tissue supplies anti-apoptotic signals into the cells within that tissue. As the tissue becomes unhealthy or the cells aren't doing their job, then that's when the anti-apoptotic signals go away, which then allows apoptosis. Now, there are other extracellular signals, so the granzymes that we've already talked about, but there's also something called the FAS ligand. And when we look at it, really, who cares about the biochemistry of this at this point? Basically, this ligand, this signaler, will will go to an FAS receptor on the cell, which will then activate a cascade of biochemistry that causes the cell to undergo apoptosis. This is what it ends up looking like. This schematic will help you kind of understand a little bit. So you've got cell injury. So not enough growth factor, DNA damage, free radicals, toxins, radiation, bad proteins being made, the wrong pH. All of these things damage the mitochondria and other things, which then initiates the caspases, which then cause the DNA to break up, which triggers apoptosis. Or this extrinsic pathway that says, okay, you're done, and you end up with apoptosis. The takeaway from this is that what happens here is that the nuclear membrane starts to form these blebs, these bulges, with lots of contents of the cytoplasm in there. These break off into what are called the apoptotic bodies, and the apoptotic bodies get phagocytized by the macrophages, where they're broken down and their components released and reused. So the phagocytes clean this up. But at no point does the cytoplasm end up spilling out into the interstitium and cause inflammation. So these are internalized because it's kind of vesicular transport almost. So a very common test question is to compare and contrast apoptosis to necrosis. You have a normal cell, it gets injured. If it can recover, it recovers and it's back to a normal cell. If it reaches the tipping point, the injury gets progressive and it can't recover, then the cell membrane starts to break down. All of the internal stuff is disrupted. The organelles are not good. The nucleus breaks apart. The contents leak out. It causes inflammation. That's necrosis. Who knows at what point this reversible injury becomes the irreversible injury, but it goes to necrosis. In apoptosis, the normal cell, the anti-apoptotic signals decrease, apoptosis happens, the cells fragment into apoptotic bodies, the apoptotic bodies are phagocytized, no inflammation. Okay, another important injury, and we're almost to the end here, an important injury to cells are proteins. Now, proteins, in order to function properly, need to be folded correctly. They need to have their tertiary structures in a very specific way. Now, if the protein amino acids are all put together properly, but the protein is malfolded because of stressors, because of pH imbalances, or because of whatever, then that protein either needs to have to be refolded properly into its proper 3D shape, or it has to be removed. If it builds up, then the cell will die. So there are these proteins that are in the cells that are called the chaperone proteins. Sometimes they're called heat shock proteins or stress proteins. I learned them as chaperone proteins, and that's the name that's most commonly used. Chaperone means it takes care of you. It kind of is the babysitter, right? So chaperone proteins bind with the misshapen proteins and reshapes it properly. If the chaperone protein doesn't work, if it can't make this protein go back to its correct shape, then another protein called ubiquitin binds to that protein and targets it for removal. And when it's bound to ubiquitin, then proteasomes, which contain proteolytic enzymes, take over, and the protein is broken back down into its amino acids, and the amino acids can be reused. And this is preventing injury to the cell. So you have proteins, various stresses and things cause them to misfold. And a couple of things happen when this happens. One of the things is you stop making them because you don't want any more of these accumulated. So you slow down on the translation of these proteins. You make these chaperone proteins that try and repair. If you can't repair, then the ubiquitin binds to them, goes to the proteasome, degrades it into the amino acids that can be used. These things are reversible. If there's too much accumulation, then the caspases will activate, and it'll trigger apoptosis, and the cell will die. This is another textbook's version of how these work. So the proteins are incorrect. They get folded by the chaperone, and you end up with the matured protein in the right configuration. If it doesn't fold correctly, then you get the ubiquitin, which then goes to the proteasomes, which then degrades it and removes it. So this is all of Unit 2. The next unit will be on inflammation, immunity, and infection. A lot of that unit we did in AMP1, and I will. So we will take a closer look at that then in the next unit.